Hi, my name is Shivani Muthukumar, and my project is evaluating attribution methods alignment with deep learning model predictions in genomics. Regulatory genomics is the study of the non-coding regions of DNA and how they affect gene expression. There are many components like silencers, enhancers, and promoters that all work together and interact to contribute to the cis-regulatory network. But despite previous studies, there are many aspects of regulatory genomics that are not fully understood. But due to their many implications to many genomics diseases like Parkinson's and Alzheimer's, they require further investigation. Understanding gene regulation is fundamental in biology, and developing accurate models that can represent these complex networks is a primary objective in deep learning in genomics. CNNs and DNNs are artificial neural networks that can identify patterns in data, and recently they've become more widely used in regulatory genomics prediction tasks as they've shown high accuracy. However, these models have low interpretability, meaning that researchers can't know what's going on inside of these models and can't figure out how the model learns and how it makes its predictions. But understanding this is crucial for researchers to be able to gain insights from these models and learn from these models. To address this, attribution methods have been developed to try to explain model predictions by providing importance scores to each nucleotide in a sequence. These methods are beneficial because they can help create hypotheses about what the model might be learning by making visualizations. For example, in this sequence, this area over here with large nucleotides has high contributions to the model's predictions according to the attribution methods, and this region could be hypothesized to be a regulatory element or a binding site. However, there is no way to verify the accuracy of these methods and their alignment to the model's predictions. And before we use these methods to create hypotheses about genomics concepts, we need to make sure that we can trust the explanations that these methods give us. So the purpose of the study is to evaluate the robustness and the alignment of attribution methods with model predictions in the context of genomics models, and to investigate the reliability of these model explanations to hopefully spur advancements in the field of model trustworthiness in genomics. In this study, I focused on three attribution methods: integrated gradient, saliency maps, and smoothgrad, and I developed three tests that I ran against two models for comparison. The first being the DeepStar model, which is a convolutional neural network that predicts developmental and housekeeping enhancer activity, and the second being the EvoAug DeepStar model, which is an extension of the previous model but which is trained with evolution-inspired data augmentations that improve the model's performance. The first test that I developed is the translational robustness test. For the translation test, I evaluated how the attribution scores changed when the input sequences were translated by a varying number of nucleotides. This test helps to evaluate how robust these attribution methods are to these translations, as we're not really changing the sequence, we're just shifting it, so we expect that the attribution scores shouldn't really change. For the results, I was able to calculate the attribution method variation score, which represents how much the attribution scores changed before and after the sequence was translated. And when evaluating robustness, a lower variation score is considered better because it shows that the attribution scores weren't really affected by these translations. As the sequence wasn't really changed, it was just shifted. And in the results, we see a general trend of the EvoLog model having lower variation scores than the DeepStar model. And this suggests that the EvoLog model can provide more robust predictions and more robust attribution scores. The second test that I developed is the necessity test. For this one, I wanted to evaluate how the model predictions change when the most important nucleotides are removed from the sequence and replaced with shuffled nucleotides. Basically, the way this test works is any nucleotides with attribution scores above a given threshold will be removed from the sequence and replaced with nucleotides from the shuffled version of the sequence. The question that this test is asking is, are the nucleotides highlighted as important by the attribution methods truly important and truly necessary for the model to have high predictions? And if they are, it means that that method does accurately differentiate between the important versus non-important nucleotides. The way that I quantified the change in model predictions is by calculating the model prediction ratio, which basically represents the model prediction of the modified sequence over the model prediction of the original sequence. One thing I want to mention is that each of the three attribution methods, integrated gradients, saliency maps, and smoothgrad, they all have different metrics and values for calculating importance. And to address this, I established a set of 13 different thresholds that represent percentages of that method's maximum value. So threshold of 50 represents a value that is 50% of that method's maximum value. And in these results, we see really interesting trend lines. If you look at the integrated gradients trend line, which is the blue line on both plots, we see that at the last threshold interval from 95 to 100%, where everything above that highest threshold was removed, we see that the model prediction ratio cannot get past around 0.5 for DeepStar and around 0.3 for EvoAug, which shows that those nucleotides that are removed in that last interval are truly important for the model to have high predictions, as it can't get past a certain percentage of its original value without those nucleotides. This shows that integrated gradients does accurately identify those nucleotides as important and necessary. The trendline that are displayed by the other two methods, Salency maps and SmoothGud, that are more linear, may suggest that they're not as good at differentiating between the important versus non-important nucleotides. The third test that I developed is the sufficiency test. For this test, I wanted to evaluate how the model predictions changed when the nucleotides considered less important were removed from the sequence. So basically, the way that this test works is that any nucleotides with scores underneath a given threshold are going to be removed from the sequence and replaced with nucleotides from the shuffled version. And the question that I'm asking here is, are the nucleotides that are considered important sufficient alone for the model to have high predictions? Because if the nucleotides that have low scores don't have high contributions to the model, then removing them and changing them wouldn't have a big impact on the model. And in the results, I used the same set of 13 thresholds that I did in the last test, and I also used the same method of calculating the model prediction ratios, and we see really interesting results displayed by all three methods. We see in this first threshold interval from 0% to 5% of the max, when everything below that lowest threshold was removed, that the model prediction ratios immediately dropped from 1 to around 0.3 for DeepStar and 0.5 for EvoOg. This shows that even when the nucleotides that are only underneath the lowest threshold were removed, that it did have a significant impact on the model's predictions. Additionally, if we look at this last threshold interval from 95 to 100%, where the same nucleotides that were necessary in the previous test are the ones that are intact here, that the model prediction ratios are close to zero in both models, showing that those nucleotides that were necessary are not sufficient for the model to have high predictions, and that the low-scoring nucleotides do have notable contributions to the model's predictions. My study shows that attribution methods may not completely align with model predictions in the context of genomics models, and this poses a big problem because publications in widespread journals like Nature Genetics and Nature utilize these attribution methods in their studies as results, despite the fact that they are unevaluated and potentially unreliable to be used in the genomics domain. The tests that I developed are methods that can probe the trustworthiness of these attribution methods, and they reveal the fragile nature and the disconnect between attribution scores and the model predictions. The translational robustness test showed that the robustness and reliability of attribution methods is partially dependent on the model that it's used on. This shows that different attribution methods have varying performance on different models, and this test can help researchers choose a attribution method that is suitable for the model that they're working on. The necessity and sufficiency test showed that the attribution methods do not completely align with the model's predictions, as generally it seems that they are undervaluing the contributions of low-scoring nucleotides and possibly not able to understand the involved network of regulatory genomics data. This all suggests that attribution methods may not be reliable or produce biologically meaningful explanations in the context of genomics, despite being used in biology to interpret models. For my future research, I want to evaluate more attribution methods using these tests and further explore whether these tests can be used as a metric to evaluate the trustworthiness of more model explanations. These are my references, and I thank you very much for your time.